In this last recording segment, we will discuss different classes of RNAs and their structure and function. We will start with transfer RNA, tRNA. What is the function of tRNA? tRNA represents a link between genetic code codons in mRNA and amino acid sequence in proteins. Each particular tRNA carries or attaches particular amino acid and carries it to the ribosomes, where it is added to the growing polypeptide chain. Now, we know that cells contain 20 different amino acids. However, they do contain at least 30 to 40 different types of tRNA, so there is some discrepancy here. There are more tRNAs than amino acids, so different tRNAs would attach and carry the same amino acid, and we call these tRNAs iso-accepting tRNAs, and we will talk about them a little bit more in next chapter. What characterizes structure of tRNA? Besides the common secondary structure, cloverleaf structure, it is the presence of rare modified RNA nucleotides such as ribotymine, ribotymidine, or pseudouracil. How these rare bases are created? They are not encoded in DNA. So these rare nucleotides or modified bases arise from chemical changes which are made to the 4 standard bases after transcription by tRNA modifying enzymes. Here you have one example. If you add to uracil methyl group, you will get ribotymidine. Most tRNA contain anywhere between 70 to 95 nucleotides. Some of these are complementary to each other and form hydrogen bonds. As a result is this very typical cloverleaf-like structure of tRNA. In this picture you have 3 different models of the tRNA. First is computer-generated space-filling molecular model, which actually shows 3-dimensional structure of tRNA. The second one is a ribbon model which emphasizes the internal region of base pairing, and third one, which we will discuss in more detail, is flattened cloverleaf model. Of course, tRNA in cells has a 3-dimensional structure, but in order to understand the structure and function of tRNA, we will discuss flattened cloverleaf model. So what you can see in this model, there are 4 different arms. There is some extra small arm, but 4 major arms are acceptor arm, which comprises 3 and 5' end of tRNA, anticodon arm, and 2 arms which contains the most of the rare bases, T arm and DHU arm. So acceptor arm on 3' hand and has a sequence ACC. It is a sequence which you find in every single tRNA, and it's a sequence where amino acid binds. So it's why it calls acceptor arm, because the amino acid would bind to the sequence. So this poses a question: if each amino acid is attached to the ACC sequence, how comes a different tRNA bound different amino acid? And we will talk about it a little later. What defines a sequence over all tRNA, which actually triggers different conformation within the space different 3-dimensional structure of tRNA. Anticodon arm is important because anticodon arm actually interacts with codon in mRNA, so the bases in anticodon define which tRNA with which amino acid will bind to mRNA on ribosome. Let's now talk about tRNA genes and processing. tRNA genes can be clustered or scattered. Bacteria usually have several copies of tRNA, while eukaryotic cells have many copies. Very often, for example, in human, you can find tRNA in so-called tandem repeats. In bacteria, tRNAs are usually transcribed together as one large precursor RNA, which is then cut into pieces. Also, that sequence you remember on 3' end, CCA sequence where amino acid attaches, in most prokaryote it is encoded in the gene, while in some prokaryotes and in all eukaryotes the sequences are added by a special enzyme after the transcription without any template. Now, once a tRNA is synthesized, is transcribed, it undergoes processing. And this processing basically is very different, and different tRNA groups are processed in different way. And you have right here only one example of one tRNA, how it is processed. Usually there is a splicing. Most of tRNA contains introns which are spliced but in different way from spliceosome mediated processing. Ribosomes and ribosomal RNA play a very important role in the translation of the genetic information contained in mRNA into the amino acid sequence of polypeptide. Ribosomes are highly abundant in the cells, in bacteria, and also in eukaryotes. Ribosomes typically contain about 80% of total cellular RNA in eukaryotic cells, so it's highly abundant. Ribosomes are formed by ribosomal RNAs, rRNAs, and proteins. In both bacteria and eukaryotes, ribosomes are formed by two subunits, large subunit and small subunit. Large subunit is formed by more than one RNA and proteins. In bacteria, large subunit, which is 50S, is formed by 23S and 5S rRNA. In eukaryotic cells, a large subunit is formed by 3 different ribosomal RNAs. Now, how do we measure? Did you notice S? S stand for Swedberg's unit. Swedberg's units measure speed by which object sediments within the centrifugation field. And Swedberg units, S units, are not additive. The combining, for example, large and small subunit will not give you the total size of ribosomes. In eukaryote, total ribosome size is 80S, while large subunit is 60S and small is 40S. The genes for rRNA, ribosomal RNA, like those for tRNA, can be present in multiple copies, and this number of copies can vary between species. In eukaryotic cell, usually you will find the ribosomal RNA genes in tandem repeats. Once these genes are transcribed, they undergo processing. Let's start with the prokaryotic RNAs. In bacteria, this RNA gene is transcribed all 3 RNAs, you remember. In bacteria we have 16S, 23, and 5S. All of them are transcribed into one long precursor RNA of 30S. This precursor RNA is afterwards methylated, and the methylation triggers its cleavage into intermediates, which are afterwards trimmed and yields the mature molecule. The process in eukaryotic RNA is very similar. It first starts as precursor RNA, which is methylated, and afterwards is spliced. But the splicing, this cleavage, is mediated by small nucleolar RNA, SNO RNAs. And the small nucleolar RNA, interestingly, some of them are encoded by sequences which you can find within the intron. All processing of ribosomal RNA and ribosome assembly take place in the nucleus of the eukaryotic cell. We will now discuss small RNA molecules which actually are involved in different functions in cells. And we will start with RNA interference. RNA interference is very powerful mechanism in eukaryotic cells which limits the invasion of foreign genes such as viruses and transposomes. RNA interference was observed first time in 1998 by Andrew Fire and Craig Mello, who were studying gene silencing in nematodes. They come up with very surprising discovery that injection of double-stranded RNA had much higher potency in silencing genes than injection of single-stranded RNA. Nowadays we know why. It's a double-stranded RNA which actually is processed into siRNA or microRNA, two RNA, small RNA molecule which are involved in RNA interference. Now, for this discovery, Fire and Mello obtained Nobel Prize for Physiology and Medicine in 2006. So, what siRNA and mRNA do? They can interfere with translation. They can bind to mRNA and trigger degradation of mRNA, its cleavage, and also siRNA can interfere with gene transcription. In addition to small RNA, siRNA, and microRNA, there is third one which you can find in eukaryotic germ cells, which is PIWI RNA. PIWI RNA particularly targets a transposon and inhibits the function of the transposomes. Bacterial cells or bacteria do not possess micro and siRNA. However, they have very similar mechanism to RNA interference, which is mediated by a CRISPR-Cas system, and we will talk a little bit more about that. Both microRNA and siRNA are produced from double-stranded RNAs. However, difference between them is from where that double-stranded originates. Usually, siRNA, this double-stranded RNA originates from the viruses, transposomes, and form long hairpins which are cleaved. MicroRNAs are usually transcribed from genes where you can find inverted repeats which forms a short hairpin structure. Nevertheless, in both cases, it's double-stranded RNA which is cleaved by dicer. Okay, dicer removes the terminal loop in hairpins, and dicer also cleaves the long double-stranded RNA, and one of the strand of this double-stranded RNA which was cleaved is degraded, and one associated with complex of proteins, so-called RNA-induced silencing complex or RISC. In both cases, for siRNA and microRNA, it's similar. Now, RNA, which is part of that complex, pairs with target sequence, and proteins which are associated either cleaved that complex or trigger degradation. Now, microRNA basically can pair with target sequence which is not 100% complementary, while siRNAs requires complementary target sequence. While siRNAs are considered as a defense mechanism of the cells against the invading molecules, microRNA actually plays very important role in the regulation of gene expression. MicroRNAs have been found in all eukaryotic organisms, and they control expression of genes during the development, growth, many biological processes, and also they are involved in control of metabolism. Most microRNA genes are found actually in regions of so-called non-coding DNA or within introns regions. So in previous slide, we saw that there are many similarities how micro and siRNAs are processed. So there are really many similarities between them. However, there are also many differences. Let's start with similarities. Both siRNA and microRNA are made by approximately 21 to 25 nucleotides. So the length is between 21 and 25 nucleotides. Both of these RNA, siRNA and microRNA are involved in degradation of mRNA, and both can be involved in chromatin modification. However, siRNA can also inhibit transcription and microRNA translation, and as we mentioned before, microRNA plays a role in the control of gene expression of many different processes in the cells, while siRNA is mostly regarded as a defense mechanism of the cells. Now, what are the differences? The origin and what they target. Origin of siRNA is mostly from mRNA, transposon, or viruses, which form RNA duplex with long hairpins, while microRNA is transcribed from genes within genome, nuclear genome, and both this gene forms short hairpins because they have inverted repeats and form short hairpins of double-stranded RNA. So both form double-stranded RNAs, but origin of this double-stranded RNA and length is different. Also, what is different is what they target. siRNA usually target gene from which they were transcribed, while microRNA target genes other than those from which they were transcribed. As mentioned before, also prokaryotes, bacteria, do not possess interfering RNA, siRNA or microRNA. They possess CRISPR RNA, CRISPR-Cas system. This system functions as a defense against the invading DNA bacteriophages. And CRISPR-Cas system, why we mention it now, is because it is used by many research laboratories to target human genes, so in genetic engineering. And what it is, for basically in bacteria, they have a so-called palindromic sequence within which they have interspersed region between those palindromes. Whenever a bacteriophage or DNA pathogen invade bacteria, you remember bacteria has as a defense mechanism restriction enzymes. Those restriction enzyme cleaves those that invading DNA, and actually part of those DNA can be incorporated within this palindromic sequence. So whenever that pathogen, that bacteriophage infect bacteria again, this CRISPR target sequence is transcribed, cleaved, and it complex with so-called Cas9, which is DNase, which is enzyme, and basically upon pairing of this CRISPR RNA, which was previously incorporated into genome and was transcribed in that CRISPR RNA, upon its pairing with target phage DNA, that Cas9 DNA would digest and cleave that DNA. Now, this is a very smart system in bacteria, which we consider as an immune response against invading pathogen. Now, what I would like to summarize at the end is, or mention, is that besides small interfering RNA, microRNA, pvRNA, in eukaryotics we have so-called long non-coding RNAs which regulate gene expression. Actually, about only 1% of the human genomes codes directly for proteins. Over 80% of transcribed RNA which are produced are so-called long RNA molecules which do not code for proteins. They do not have start and stop codon, so they do not have open reading frame. for encoding proteins. And basically, the region from which they are derived are called something as a dark matter of the genome, because we didn't know really for a long time what these long non-coding RNAs are, what they do. But we now know that they may regulate protein expression. One of these long non-coding RNAs we already discussed, you remember, zized RNA, which play a role in dosage compensation in mammalian cells, which is transcribed only from the X chromosome, which is designed to become inactive. So, besides zized, there are many other long non-coded RNA, non-coding RNA, which play a role in gene expression regulation. Some of them actually can be involved in genomic imprinting as well.